So we just finished up glycolysis and a little bit with fermentation. Now we are going to start talking about the Krebs cycle. But before we can get there, we have to talk about the transition step. And so we take those pyruvate, we're going to talk about how it turns into acetyl-CoA. So this is starting on page number 51 in your questions. It says look at figure 6.10. And so you have your 3-carbon pyruvic acid, and carbon dioxide is being removed from the pyruvic acid, and coenzyme A is added and changed into acetyl-CoA. So we took from 3-carbon, we took a carbon dioxide off, which is called decarboxylated, and turned into a 2-carbon acetyl-CoA. So where is this happening? The mitochondrial matrix. We've talked about this several times. So kind of the cytoplasm-y stuff in the Krebs cycle, in the mitochondria. So the part of the Krebs cycle that you guys are really going to have to know is this beginning part. Acetyl-CoA, just 2 carbons, and that came in from the transition step, bonds with acetyl-O-acetate, which is 4 carbons, to make the 6-carbon citrate. So 2 plus 4 equals 6 carbons. It takes 2 turns of the Krebs cycle for every glucose. So why would that be? So if you remember, our glucose broke into 2 pyruvates. Each of those pyruvates take a turn around the Krebs cycle, which is why we have 2 turns. So what's happening? It requires oxygen, so that's aerobic, and there's going to be a whole bunch of oxidation reactions that give off CO2 and produce 1 ATP per cycle. So we're getting a total of 2 ATP total. So you've almost been able to answer all your questions just with that. So again, we have our acetyl-CoA coming in from our transition step, meeting with our oxaloacetate, our 4-carbon, to make citrate. This is just the general scheme. We're going to go into more in-depth one. But in general, what's happening is 2 carbon dioxide are given off, 3 NADHs. This is per turn. This is only 1 turn here. 1 ATP and 1 FADH2. So if I did 2 turns, I'm going to get 4 carbon dioxide, 6 NADHs, 2 ATP, and 2 FADH2s. So a little bit more in depth. Excuse me. This is the step you guys really need to know. Right here, I have my acetyl-CoA, which comes in again from the transition step. Look, I have 2 carbons. You can count them up. It's going to join with the oxaloacetate. So I have my 4 carbons right there to make citrate, which is also why it's also called the citric acid cycle. So if you remember the cell respiration song, this is where it rearranges. And so we call it different names. You do not need to know these names. You do not have to memorize these names. I just want you guys to see it and see what's happening. So notice I still have 6 carbons here if you count them up. Look, decarboxylated. I'm taking a carbon dioxide off, and if you count, I have 4 carbons. Change the name. Do it again. I lost another carbon. I'm down to 4 carbons. Change the name. Also at the same time, I'm making my NADHs. I made an ATP right here. And rearrange, new name. Rearrange, new name, made a FADH. Rearrange, new name, added some water in. I got another NADH. So this is the Krebs cycle. Again, the parts you really need to know are up here. So in total, after 2 turns, you're going to have 2 ATP, 6 NADHs, 2 FADH2s, and 4 CO2s. And you can see that through all the different parts. That is it. This is just another part that shows everything all together.